Hello, everyone. I'm showing that we still have about a minute before we get started. We'll proceed at that point. All right, well, my computer says it's 10 o'clock, so I think we'll go ahead and get started. I'd like to welcome everyone to the second half of Maintaining the Taxonomic Backbone, or Connecting Those Who Try. I'm Teresa Mayfield-Meyer, and I'll be your moderator for the session. My co-moderators are Vijay, Barve, and Nick Dowdy, and we're grateful to the tech support from the University of Florida conference staff. Before we get started, I want to remind everyone that the session is being recorded and to ask speakers to speak slowly and clearly. Attendees, please post your questions in the Whova Q&A application. This will allow others to vote up your question, and any questions that don't get answered in our sessions can be answered and reviewed later. If time permits between presentations, we will address presentation-specific questions. Otherwise, we will hold questions to the end when we have time for discussion. Finally, we ask that everyone please bear with any technical difficulties we may have and that everyone follow the conference code of conduct. We hope everyone had time to digest yesterday's talks, and we're sure that today's presentations will build upon yesterday's discussion. So let's get started. Our first speaker will be Emilio Berti, postdoc in the theory group at IDIV. This talk is Managing Species Names Across Biodiversity Databases, Sources, Tools, Pitfalls, and Best Practices for Taxonomic Harmonization.
I just share my screen. So welcome everybody, and just to introduce myself a bit, I'm not a taxonomist, but I'm a macroecologist. And what macroecologists do is that they get several data sets and they're trying to combine them together to ask some question. And the way to combine them together is through taxonomy. So one of the problems we face is taxonomic harmonization. And I have to say that we work on it because we have to. We are not particularly pleased to work with, and all my talk would be from a user perspective more than from a developer point of view. So let's say I have dataset A and B, and taxonomic harmonization is needed because if I want to combine these two datasets, I need to know which species in dataset A is actually the same in dataset B, even if it's called with another name. So taxonomy is central for the work of macroecologists, and the way I used to harmonize taxonomy was using GBIF because they have everything and it's fast and it's easy to do. When I moved to IDIV, I started talking with people here, and I found another guy, one who's actually doing exactly what I was doing, but using another database and using another framework. And we started talking about this, and we end up being 5 people who exactly wanted to do the same thing, but each one of us is a different approach of doing it. And so we start working on the problem, and this is how this talk came to be. Is there one best way to harmonize taxonomy? That was our $1 million question. And yeah, we went down a bit of a rabbit hole that was very interesting. And my talk is divided into two parts. What are the available resources? So I'm talking about databases and tools, but only our packages because that's what ecologists mostly use. And then which workflow is most suitable for some specific tasks. Start with this. So we compile a table with all the database we could find, which is also summarized in this figure. So on the x-axis, taxonomic breadth of the database, and on the y-axis, the spatial scale. And on one extent, we have a regional database that focus on one taxonomic group, for example, German SL, which is for plants in Germany. And on the other extreme, we have global databases that have no taxonomic restrictions, so try to include all living beings like catalog of life and GDWG. Once we map the database, we start looking for all our packages that we can find on CRAN and bioconductor, and we divide it in this category to access the resource and wrangle data and visualize it. From these two tables, we combined into a bigger table, and we made a Shiny application that is available at this website through this QR code. It is composed of two parts. The first part is the table where you can search for a string, and it will pop up database of packages to work on this taxa with some information here. What most interesting is interactive network visualization of this table with databases which are triangles and the relationship between them. So for example, in this case, Tropicos database is used to populate GNR, and with the circles are the packages that can be used to access the databases. And this is useful for users and people that are not particularly trained in taxonomy, because if I want to work with several databases, I can just click on them and I know which package to use. Once we made this roadmap, we start focusing on how to actually harmonize a list of species names. We needed we just needed a list of species to work with as an example, and we choose a biotime database, which is particularly interesting because it has a large spatiotemporal scale. It's a database containing information of species composition turnover over time, and it was compiled from many sources that probably use different taxonomic authorities, so we expected taxonomy to be not so internally coherent compared to other more curated databases. We define three workflows that are philosophically distinct, that I'll try to summarize very briefly. We always start with a raw species of list from the database biotime. We standardize, meaning we drop author, year, and everything else that is not genus species, which is not the best approach if you ask taxonomists, but we are not, so we do not care. The first workflow just gets the list and pass through the taxonomic database of interest, in this case for plants, birds, and fishes, and just pass the whole list to each of these one. And we end up with three lists that are then merged, resolving conflicts between the list, and we end up with an harmonized list of species. We call it this taxon-specific because we use taxon-specific databases, but no groups, just pass everything to everything. The second workflow is quite similar to the first, but we have a first step in which we use a global multi-taxa database, in this case GBIF, to split the species list in higher taxonomic group. And so we make three sublists that are passed only to the specific database of interest, and we end up with three lists that can just be concatenated together to get an harmonized list of species names. And finally, we call this taxon specific because it's still using taxon specific database for the final queries, but it's grouped using a multi-taxa database. And finally, we just use GBIF, which is the approach I did in the first place, and it's fast, easier, and I would say is the one that most appeal macroecologists and people that are not used to work with taxonomy. This is just a multi-taxa database workflow. It's very fast and easy to do. These are the result of these three workflows that are on the columns, and the numbers show the number of species that were identified in each of the three classes that we decided to study. And if we just see workflow number 1 and 2, they're almost identical, except one species, birds and fishes, that are not matched here, and some plant species that are lost here. And we know this difference can come only from the fact that GBIF sometimes assign to a taxon that in reality is a plant to something else, so this is simply not passed to the database. But the biggest difference is between the workflow number 3 that uses only GBIF compared to the 2. GBIF always matches the highest number of species, but it doesn't resolve many of the synonyms that are resolved in the 2 workflow. So, which inflates this number here, and it's not so clear what's going to happen regarding taxonomic hypothesis. Summarize a bit this part. The first workflow is the most accurate, but it's quite slow and complex to implement. It will match the most species accurately, but you cannot do periodically, let's say. The second one is good enough. Actually, the accuracy is only marginally worse than the first one. It's fast to implement, it's also easier to implement, but relies on this first step of multi-taxa database, so splitting into groups. Finally, the third is the fast and easiest, as you know, but miss many synonyms and possibly mix taxonomic hypotheses. Going to conclude with 3 take-home messages and taxonomic database tools in the relationship for me and macroecologists and users that are not used to work with this information. It looks something like this. It's a very complex landscape, and we have no idea where we are. We have no idea where we want to go or how to get there. And in this sense, our shiny update is still partly incomplete because some of this information, for example, of the link, is a bit hard to find, but we are still working to be as exhaustive as possible. This app provides a roadmap to navigate this complex landscape, and for a user, it can be used to go very quickly to decide what database and tools to use. Finally, we highlighted some pro and cons of each workflow. In our case, for harmonizing biotime, we think workflow number 2 is the best, but we also discovered with our great disappointment that there is no silver bullet to harmonize taxonomy, and we should really, whatever works for you and for your task is probably change every time. We hope to have highlight some of the different results that come with a different philosophical and conceptual attitude to the problem. This I would like to thank you all and the organizer for having me my support.
Thank you very much, Emilio. The Escher staircase is so apt. I love that. So, do we have any questions for Emilio?
None yet.
I'll just say, so Nick and Vijay and I are kind of familiar with what you just did because we did a lot of that as well for different groups. And I think I like the way that you presented this with the 3 different methods of getting there and how they compare to each other. Because I think we felt a lot of frustration with, there's all these resources, what are we supposed to use and how do they overlap? So it was refreshing to see that somebody else was experiencing the same thing we were.
All right, we got some questions.
Cool.
So first one is, how was the matching done? Was it based on a string match?
Yes, so the species list, the raw species list is just genus species and something. So we did not know how to work with not working with string match. So yeah, pretty much.
Okay. Next question is, how do you process or handle homonyms?
Well, it's a good question. So we rely on these. I don't know. I don't process them. We just trust the taxon-specific databases. Like they should get the information. In this sense, we hope things goes well. But maybe I misunderstood the question. Like homonyms between the different classes. Yeah.
They've given an example of basically like a taxon that has the same name but a different reference, for example.
Yeah, so that would be different like taxonomic authority, right? Yeah, we drop everything and we consider it the same species, and that's it. If something appears in two, like if they call the same name fish and plants, and there is no other information, the safest thing to do is to omit the species. But I think a good point that I omitted here is that the string comparison are done sometimes by fuzzy matching. And that actually open a lot of opportunities for sensitivity analysis that I never saw a lot of them, but it's a possibility.
Okay, we have one more. Did you factor in that some of the scientific names in biotime might have already been through a name harmonization process? For instance, data sets that come from obis would have been matched to worms.
No, I mean, we did not think about it. I have no idea what that implies. If someone can tell me, I will be very happy.
I have a question for you.
Yeah.
Did you, through this process, did you find that like certain taxonomic groups have a lot of resources that need to be harmonized and integrated versus others that have maybe no resources at all other than kind of the general big resources?
Yeah, I mean, for that's the classical bias of macroecologists. We have most of the data is for a fraction of the biodiversity, and that's true also for taxonomy. I mean, we biotime has many species. We focus on birds, fishes, and plants because it's the one we could find the databases and tools to access them. But like, if you want to, at some point we thought, let's try to harmonize some order of insects, and at that point you have to rely on multi-taxa databases, global one. I mean, that's the only way. No.
Excellent. All right. Thank you, Emilio.
Thanks to you.
Our next presenter is Laura Rocha Prado. She's the biodiversity informatician and full stack web developer at Arizona State University, the Symbiota Support Hub. Her talk will be a simple set of tools for assembling, annotating, versioning, and publishing taxonomies.
Thank you for having me. My name is Laura, and I'm a biodiversity informatician at Arizona State University, and I'm also a full stack web developer. And for the past few years, I have been collaborating with the Symbiota Support Hub to develop new tools in the Symbiota software. Today, I want to talk to you guys about a set of tools that I'm developing to help the independent assembly and publishing of taxonomic classifications. This has been a passion project for me for quite some time now, and I'm really happy to finally introduce that to you. But first, I just want to do a brief background on why I created this. Well, I'm a South American leaf beetle taxonomist specialized in neotropical beetles. And as I'm sure all of you know, taxonomy is really hard, especially if you work with hyperdiverse neotropical groups. The taxonomic history of these groups is often complicated, hard, or even impossible to track. There aren't enough digital resources to help, and type specimens are often inaccessible. If you're a scientist in training or someone starting with a group like the one I study, you have to rely on ancient publications. There are no other specialists to help you. There are no online faunas to look. no catalogs, no manuscripts, and sometimes the names aren't even available anywhere. You're dealing with what I like to call the dark biodiversity data. Data that exists, but isn't discoverable online. To a lot of aggregators, if it's not online, it doesn't exist. However, if you want to pioneer the digitization of some taxa, you're in trouble because you have to first create a taxonomic backbone from scratch, since all the software like Symbiota or Specify require a taxonomic backbone for you to place your digitized specimens. So thinking of these users and thinking that if I'm not a member of a thriving community of taxonomists with no funding, how can I contribute to existing taxonomic backbones if I'm not a member of those communities? Well, that's why I wanted to create this tool. Here are a few use cases that I hope to implement. Thinking of someone that wants to make several published taxonomic trees available online for both humans and computers because they're doing a sort of taxonomic alignment study where they want to compare different taxonomic classifications and their change throughout time. Or think of a user that wants to take a snapshot of a version of a taxonomic backbone using a Symbiota portal. Or maybe someone who wants to establish a Symbiota portal but needs to import a taxonomic backbone from scratch. And they want to create one because they can't find one online. Thinking of all those use cases, I thought of this architecture for the set of tools. This diagram illustrates how the tools interact and their roles in the architecture. On the left side here, you can see the curator tool, which is the front-end client and the website that is visible to users, where they can then assemble taxonomies and export them to the data sets or files. Or they can save them to the aggregator. The aggregator is the backend and is responsible for making the bridge between the client and the database, and also serves as an API to export data from the database to computers. This is a screenshot of a functional prototype that I'm developing in React.js, which is a JavaScript framework. And I just wanted to showcase how user-friendly and simple I want it to be. There isn't much to it, just an interface where you can, for now, just create and save new taxonomies. This other screenshot is from an early mock-up of the curator tool, where you can see there are 3 modes to create a desired taxonomy. The manual editor, where users can drag and drop scientific names from a search tool, a harvest from URL or external sources, or upload a CSV or a spreadsheet. Harvesting from external sources will be possible at least with Symbiota portals, because I'm developing a module for versioning Symbiota taxonomic backbones, which are also known as central thesaurus. And integration with major aggregators is also envisioned as soon as I can study their data models and their available open APIs. So it would be potentially as easy as picking an online source to just grab a copy of their taxonomic backbone and modify it and annotate it if desired. So this is another early mock-up of the curator tool, where users can search for a particular taxon. And this search would be powered by the existing aggregators, like the Encyclopedia of Life or Catalog of Life, ITs, and so on. And they could then either select existing taxa or create their own and add it to the desired tree. Once a user saves a taxonomy, they can then export it to a variety of formats. And I want to introduce as many formats as possible to help taxonomists in different contexts. So for instance, if you want to export your classification for computers, you can pick either JSON or XML. Or if you are looking into publishing a taxonomy in a paper or your website, you can pick something like the HTML format or the rich text format. or you can even export your backbone into a Nexus compliant software or Delta software to improve descriptions or even to provide systematic information. The aggregator tool is completely optional and independent. It's basically the application connecting the front end, which is the curator, with the database. And I'm envisioning some sort of user authentication layer so that users can publish taxonomies and then have unique identifiers that can be persistent and long-lasting. The aggregator would also serve the curator tool for searches. So someone arriving at the curator tool could then look for existing taxonomies before creating their own. This is just a screenshot of one of the JSON endpoints that I'm envisioning for the aggregator tool that I modeled in a Node.js prototype. Node.js is also JavaScript technology. I'm using in my data model Darwin core terms whenever possible. And other terms are going to be explained in a data model that will be made available. And I'm envisioning a few data points in the API for users to see all taxonomies, a single taxonomy, or to query taxonomies by scientific name, for instance. At the current moment, I'm developing a minimally functional prototype that I'll be posting on this GitHub repository that you can see in the URL in your screen. And since I've been doing this in an independent manner, I'd like to make it as collaborative as possible. So I'm asking anyone interested in helping me with any sort of contributions, from suggestions and questions to code, if you're able to. I'm asking you to just interact with me on GitHub in the discussions tab. This is what the discussions look like in GitHub. It's a relatively new feature where anyone with a GitHub account can post a discussion and start a topic. And those discussions can eventually become issues and even guide the development of the tools. Because this is very experimental and is still in an early development stage, I really want to hear your thoughts. I'm also getting in touch with developers for biodiversity tools to leverage their open data and maybe establish some sort of collaboration, because it doesn't make sense to do this in a bubble. I hope to publish an early beta version soon. I'd like to thank everyone. If you'd like to build this with me, please let me know. I'd like to thank the conference and the symposium organizers and everyone who reviewed my abstract and everyone who gave me suggestions, encouragement, or became a beta tester. Thank you all, and I hope to hear from you.
Thank you, Laura.
Encouragement or became a beta.
Oops. Sweet. All right, Vijay, do we have any questions?
No, I do not see any questions yet.
We've got one now.
Okay, so we have a question coming in. Why not publish your checklist using a Darwin Core archive as you suggest them to submit to Catalog of Life and call it back from the service? Then everyone benefits from filling the gaps.
Yeah, that's a great question. And it is something that I'm envisioning, the ability to just directly publish to the desired aggregators, Catalog of Life or ITIS even, or GBIF, anywhere where you could use a taxonomic classification. Yeah. I just want to say that the idea is to create a platform that anyone can build a taxonomy and because I sense there's some gatekeeping in the taxonomic community where if you're not a member of a thriving community of taxonomists, it's virtually impossible to contribute. I know people say their taxonomies are open for contributions, but that's not really the case in my experience at least. And so, I had to work around and build my own solution because I wanted to like join the team. So this is for everyone that feels left out.
Yeah, right. I think
Same here.
Nick and Vijay and I can totally relate to that. So, and I do think, I mean, even if you do have Darwin Core archive exports, there are still barriers to getting that data into some of these, especially the large aggregators. So that'll probably be an ongoing conversation over time, I think.
We have one more question. How do you handle edits? The Australian plant name index software has some complex tree delta system.
Yeah, this is something that I'm still figuring out because the idea would be that once a taxonomy is published, so pushed to an aggregator or the API, it should get a DOI or some sort of unique identifier and version, right? So I'm thinking maybe using Zenodo to keep track of versions so that you don't lose the history. Otherwise, I wouldn't allow for edits after the publication or the issuing of a identifier just to avoid confusion. You have to keep track of the versions somehow. So for now, the simplest way of doing it would just to have a database to keep the temporary trees or while people are editing it. And once they're published, then that goes away from the system I'm building and goes directly to the aggregators or the places where people need them. So then it's up to them to deal with the versions that are published. So, yeah.
Okay, there is one more question or comment. Some of the things that you want to do is you want to do are possible by using scratch pads.
Yes, I agree. But scratch pads has a barrier to get in. I have a scratch pads account. I don't want to offend anyone, but I don't think it's the most user-friendly system. And I've worked with a lot of taxonomists in different areas, and we have to make something more user-friendly so that really anyone can edit a tree. And they don't even, in my opinion, they shouldn't even know what Darwin Core is.
Right.
They should just be able to produce something that is Darwin Core compliant, but it's not necessarily, they're not like professionals in Darwin Core or data standards at all. They should be taxonomists, specialists in taxonomy, not necessarily data standards. That's just my opinion. So that's why I wanted to make this, and I want it to be very user-friendly, and I don't mind to be not like a unique tool. I just want it to be accessible. I think the more the merrier, to be honest. So.
Excellent. Thank you, Laura.
Thank you.
Okay, our next speaker is Matt Yoder from TaxonWorks. His talk is Nomenclature Over 5 Years in TaxonWorks, Approach, Implementation, Limitations, and Outcomes.
Hi, everyone. A special thanks to the organizers for putting this together. It's really wonderful to have sort of the collective brainstorming that's going on here shared with everyone. You can all see my screen and hear me, I think. So I want to mention that I am also a biodiversity informatician like Laura, and I'm fortunate enough to be part of the species file group. This is an endowed group with many wonderful people that have a lot of wonderful expertise. I won't call out all their expertise, but I think if you look here deeply, you'll see TDWG, global names, catalog of life, world-class systems administrators, we have professional designers, we have great biologists. And so really what I'm presenting here is a reflection of this team. So all the mistakes are mine, all the cool things really belong to this group. So as most TDWG talks go for me, I'd like to start with a bit of philosophizing and think about backbones in general. I was trying to think about what does it mean to create a backbone. And I think there's a bunch of different kinds we can think of. We could think of phylogenetic backbones, quantitative phylogenies, natural backbones. So these are where the nodes are biological species, for example. And they may just be taxonomic assertions by a taxonomist, and they may be mixed with phylogenetic assertions. We have a nomenclature backbone, and that's really one slice of taxon works that I'm going to focus on for this talk. And these are strictly sort of more objective. They're a recording of facts, and I'll try to explain a little bit of that. But really, I think when we get to things that we think about as backbones, they're sort of staging platforms for letting us do work. And these staging backbones are a mixture of all of these different types of classifiers or classifications. So a little bit more abstractly, here in the middle, we see a number of backbone types intertwined. And we can observe that maybe a backbone strength grows as data are integrated across it. Or is the backbone the collective integration? Is there one data structure that is the backbone? It's an interesting question. And so I mentioned that I'm going to zoom in here on the green line on the nomenclature line. And if we get right down to looking at the data, we almost have a sort of looking at a microscope in a pool of water where we can see many different kinds of organisms. This is strictly nomenclatural data and the relationships among names and the statuses of names here. You'll notice over in the bottom right on the target, there are some other structures that look to be a little bit more complex. And if we zoom out and run a classifier on a GraphViz graph of nomenclature, we can start to see a richer, more complex sort of structure. Here's another view of that graph. You can look at the depth of the nomenclature that's happening here. If we zoom way out and look at the collective data set that we have here, we can sort of wonder what this is. This is Dmitry Dmitriev's 3i Okanarenka nomenclature. There's around 15 years of research on this. This is nomenclatural facts only. So by that, I mean, if I gave you this, you would not be able to say anything about the biology of what is going on in this data. There's around 1 million rows. There's many different edge types. This was scripted off of our JSON API. And for those of you who are envisioning like the broader integration, it's important to note that almost 80% of the citations on this graph are older than the year 2000. So while Plazi is a wonderful solution for upcoming work, we still have a lot of historical effort that needs to be captured. So how did we build out the nomenclature and how did we build out taxon works? There's almost too much to talk about, so I'm just going to hit a couple of abstract points. We talked to, as other people have mentioned, the curators, taxonomists, and systematists, and we looked at their databases and we spent a lot of time with them, many years. Perhaps the biggest thing I can sort of pound home is that we did not confuse biology and nomenclature. So if your database has a name and you have a direct linkage to a collection object, a physical specimen, then in my opinion, you're not doing it right and you're going to have some problems, and we can get into that. We thought about modeling things, not rows or strings or fields, and we treated the models, the things that we modeled, all equally. So by this, if we can annotate one thing with an image or a note or an alternate value, a spelling, then we can annotate all things in our model. It's also important to note that we speak standards, but we're not constrained by them. So by that, we mean we didn't create a system that has the Darwin core as the model, for example, or a JSON API that is JSON LD or something very specific. We can export to that, but in our application development, we're not constrained by that. I've talked a lot about the nomenclatural model, and I don't have time. We have a formalization that came out of it. We talked about it earlier. There's links at the end, but essentially we have a graph, as you saw in the image there. And everything in the graph you can notice can have, just as I mentioned, citations or annotations on it, all of the different parts, the nodes and the edges on it. And I also don't have a lot of time to talk about implementation, but I did want it to just pop up one single screen. This is a view screen, not as an edit screen, to give you an idea of the kind of things that we've thought about: custom attributes, data validation, quick navigation with radial annotators, project management, where you can create your own controlled vocabulary terms. And as I mentioned, you can annotate everything equally. So here's a taxon name record, a class of things that are taxon name, and they too have images. But what's an image of Orniotes? It's not of the taxon, it's actually of the name. This is a LEP index card catalog. We have some 500,000 of these that we've grandfathered into Taxon Works. So we've hit a bunch of big limitations, and of course there's many, as Laura pointed out. And I just want to tap on a couple of those that I think are maybe useful to think about in a group context. We've been meeting for well over 2 years now on a weekly basis with our community of users and other developers, and we've accumulated a lot of help documentation, and we really need to unify that and centralize that to make it easier to get on board with Taxon Works and the other tools that we're developing in the species file group. We've had many discussions and we've seen thousands of hours of use of Taxon Works, and it's very clear to us that things that Laura alluded to, the need for these standalone interfaces that sit on top of some of the big APIs are going to be critical as sort of intermediate components that link you into Taxon Works. And in particular, we've met with Plazi several times, and it's clear that their parsing of the current literature will make things a lot easier for those who have complete data sets like Dimitri has. Largely, he's captured the complete nomenclature, and he just needs to keep up to date with the literature. And that's the same for numerous other very rich projects that we support. We work a lot with Darwin Core, Catalog of Life, of course. But some of our semantics, and I think this will echo true for other projects that we use, and some of the tools that we use and develop are richer than those standards. And as taxonomists building new concepts and new nomenclature and developing new data, which is a big target of Taxon Works, we want to be able to publish to the graph natively. We need to be able to come up with our own controlled vocabularies. So for example, with Nomen, we have a lot more richness that can be expressed in either Darwin Core or Catalog of Life. And it's really up to us to try to work with standards bodies like TDWG to see how we can better implement those, the work that we've done there. We've been working with the Legume group, Joe Miller and some colleagues to flesh out the botanical nomenclature side of things. I didn't mean to draw the line all the way through that, but we have a nice branch. We've made significant progress in the last couple of months to fully support the botanical nomenclature codes in our Nomen. So that's exciting for us to roll that out, hopefully within this year. And the other limitation is that users want output, as many different kinds of output as you can give them, they want more. And this is really important for onboarding other users and onboarding and growing the community to show that you can return all of that data. So just to highlight some outcomes of Taxon Works and not just the nomenclature, but the broader data, we export around almost 6% of the Catalog of Life arrives through Taxon Works now, and that with data sets in the wings, that will probably double, I suspect, in the next year or so. We've had over 14,000 hours of human curation on the project, and this is just a wealth of feedback. So getting a product out in front of users and having them be the ones to tell you that what needs to change has, I think, rapidly allowed Taxon Works to improve. Right now, we have around 40 to 50 curators that are working on average every week on Taxon Works. We've run some nice experiments where we've shared some of the data models in Taxon Works, share data across projects, and notably people and sources. And so when you add data in one project, it can get used in another project. And I did a bunch of queries, and it's really nice to see that 5,300 people, for example, are shared across projects now, and over 43,000 sources are shared across projects. So these kind of basic data in the context of Taxon Works are really sort of catalyzing the speed of what you can do and sort of the linkages. As Laura mentioned, there's a ton of different kind of outputs that we can generate. You see some on the left, and everything in TaxonWorks is driven off a JSON API, all our interfaces, etc. We have a mandate that we're for biologists and developers. We've had people that have dropped in and realized that we have autocomplete API endpoints. So Raf Frentz and the Gralnick lab has used the Odinate data and our autocomplete endpoints to work in their genomic pipelines. Jose, our developer, just built a separate autocomplete library that has a link to it. Oops, sorry, I'll dismiss this, et cetera, et cetera. And we've had websites to start being built off of our data as well. So to wrap up, I think we have, as a backbone producer or as an entity that helps produce backbones, I think we have some very nice features in TaxonWorks. We have a super rich data model with over 40 or 50 classes of things that you can do. We can annotate, we can handle images, we can georeference, we can do collections management. And one thing I'm just to highlight is that I'm very excited that we rolled in a Darwin Core import and export feature branch that should really be a game changer for letting people get data in and out of the system and integrate it into the broader backbone. So that just gives you a brief flavor of what we're doing. I'd encourage everybody who's interested and has more questions to join us this 6th to the 11th for our second annual TaxonWorks Together. You can find details on the events page, or you can contact our community liaison, Debbie Paul, or myself. We also meet regularly twice a week at minimum. You can find details on the events page. And you can find links to many of those things that I mentioned in the past at the back of the talk here. I'll share that link in the chat here in a second. Thanks, everyone.
Thanks, Matt. Do we have any questions for Matt?
Not yet, but I'm sure they're just rolling in.
Yeah.
And then just going to take the opportunity to say that anyone using Scratchpad should also keep TaxonWorks in their radars, because it's a valid alternative should Scratchpads become obsolete at some point. So it's good that we have alternatives from now.
I think Laura mentioned it already, that a diverse ecosystem of tools that can slowly share certain parts or standards or stuff really drives creativity and imagination and stuff too. So like Laura, I also don't believe in the mantra that we shouldn't reinvent the wheel. I don't think we know what the wheel is often. And so as many experiments as we can throw out for a while are worthwhile to a degree, of course.
Okay, well, we are kind of bumping up on our time frame, so I think we'll move on, and if questions come in, we can address them at the end. Our next speaker is Dima Moseran, bioinformatician at University of Illinois Champaign-Urbana, and his talk is Connecting Taxonomic Backbones Using Global Names Tools.
Can you see the slides?
Yes, we can.
Okay, so I am working on a global names tools for a while now, and we are at the second generation of these tools. So first versions of them appeared about 10 years ago, and is used in Encyclopedia of Life, BHL, Arctis, and so on, many other places. And the tools are about scientific names. So what Emilia was talking about, for example, how hard it is to work with scientific names, I totally agree about that. It is hard, and but it would be much harder if you would not have 250 years ago Linnaeus, who popularized binomial nomenclature. And tools that I am talking about are about binomial nomenclature and how to use it with these kind of names. So the projects that we have now, they are generation number 2. And people who do continue to use generation 1 tools, please consider to switch to those because we will sunset old tools at some point. And actually one of them, GNORT, which was for name finding, is already substituted with a new tool, GN Finder. So what is the difference between these tools and other tools? So these tools are much faster, and of course they're becoming better because we know more and more about what people need, about bugs that we have, and so on. So what are scientific names? Scientific names are pointers to taxon concepts. We can just point finger at a taxon concept, but that is hard. So it is much easier to give a label. And the problem with labels for taxon concepts or taxons is that they are in a dance with each other. So sometimes name appears and then disappears, but taxon concept stays. And in other cases, name stays, but taxon concept changes. And there are all kinds of different things that happen. However, taxon names, like scientific names, are pretty good proxy that allows us to aggregate data from different sources with an archaic precision. So what are scientific names? Scientific names is 100 million of name strings spread through literature, and these names are, even if they are about the same name, they differ from each other slightly. Sometimes there is an authorship, sometimes there is a year, sometimes there is a comma, sometimes there is a misspelling, sometimes people put gender wrong in the name, and all these problems they accumulate and create 100 million different name strings. But when we talk about nomenclature, nomenclature deals with about 7 or 8 million name strings or names. So how do they relate to each other? Nomenclators, they work with widely published names. And to get from this vast quantity of name strings to names, we have to put them in lexical groups. So we abstract from commas, from years. We just say, okay, all these variations, they all come to that particular name. But when you work with the taxonomic backbone, you actually work not with names. Usually you work with concepts, with the taxonomic concepts, and those relate approximately 3 to 1 on average. So on every 3 nomenclatural names, there is one taxon concept on average, and that means that when you work with taxonomic concepts in the taxonomic backbones, you have to go from this 100 million name strings to these 2.5 million taxons. So this is an example. We have a taxon that is represented here as a picture, so we can point finger on it. And there is more than 20 different name strings that all point to that particular concept. And these 20 different name strings, they are divided into 2 groups, 2 lexical groups that correspond to variety in one case and into subspecies in another case. So this was just a taxonomic decision, like somebody did at some point, and moved variety into subspecies. And somehow we have to deal with all these differences in name strings and normalize them together. So for that purpose, we have a tool. It's called GenParser. And if you put one of these name strings into the GenParser, it will return back 3 different versions of very, very normalized name. The most full one will have a complete spelling of every main part of the name, and also will have rank. The simpler one will remove rank, because that allows us to match together names without rank, names with subspecies, names with variety. And the most reduced normalized version has no suffixes, and also some letters are changed from according to Latin grammar from one letter to another. What else GenParser is good for? It allows you to check in your backbone and see how many names are well formed and how many names are not well formed. And it allows you to extract metadata like authors, years, like generic names, specific epithets, and so on. The speed is much better than old version. So on an average laptop, modern laptop, you can go with 300 million names an hour. And we try to very hard. We try to cover all the edge cases. So now our tests cover 1,000 different name strings that have one or another tricky situation in them. You can see in this link like all variants that we cover. And if there's something missing, please make a ticket. So the parser, and this is kind of our philosophy for all our tools, to have many different venues. The best is when projects like TaxonWorks or TaxSize use our tools inside and make them invisible for users. People can just enjoy the functionality. But there is also web interface, there is API, there is a command line application, and the format that we return back is either Excel friendly or friendly for more detailed JSON output. Another tool that we have is Verifier, and Verifier is a tool that uses information from more than 100 data sets, such as Catalog of Life, GBIF, and many others. And it allows you to get your checklist and see if all the names that you have are real. Are they spelled correctly? Are they currently accepted? And what other data sets have these names that you have? So we check it against 27 million different names. And I probably do not have time to go through all of it, but we have pretty much 7 different stages when we try to verify. We try to verify exact match, we try to do fuzzy match. If that doesn't work, we remove some parts of the names, and like what Laura said, dark taxa, names that disappeared from online databases but exist in the literature. For example, new subspecies. We have species, but we don't have subspecies. We have known genus, but we don't have species epithets. So we check for that, and also we check fuzzy and not fuzzy. And if everything else fails, we try to match by genus. And if it doesn't work for genus, then we say, sorry, we couldn't find anything. And when you go through all of that, you get data from these 100 different data sets. Who of them have information? For some names, output is overwhelming. For example, Escherichia coli generates 5,000 matching results, which makes it hard for, especially if you look for several names. So what we try to do, we try to return the best possible result. And one of the questions for was how you deal with homonyms. So we try to use parser data to match authors, even if they abbreviated. We try to find if this is the same authors that people meant to find. We try to find names according to how well they are parsed and how well they are they currently accepted names or they synonyms? So currently accepted names have a preference. If you don't care about all the names, you care about specific data sets, you can pick these data sets either from web interface, or if you use API or use command line application, you can use all 100 data sets and pick one of those. Output is similar. We have a similar way to install it. So you can use Brew to install command line application for that. You can use API, you can use web interface. You can use taxize hopefully soon, if it is, and the speed is about 1,000, 5,000 names a second now. I do hope it will be like 2-5 times faster in some future. Another project we have is GenFinder, and this is for finding names, but I will not talk about it much here. This is a tool that we use for BHL name index. Thank you so much for inviting. And I'm, as Matt said, in University of Illinois Species File Group. Enjoy our group very much. I think it's a wonderful, wonderful team. Thank you so much.
Thank you, Dima. Do we have any questions?
Yes, we do have a question from Yurik. Dima, thanks for all your efforts with the global names tool. I enjoyed your tools and related exchanges over the last years. Your tool provide interpretation of scientific names, parsing, finding, verifying. The question is, how can I reliably cite and reproduce a specific name interpretation you provide given a specific list of names?
This is a hard question, actually. I do not have versioning of a decision. In the sense that, for example, if Catalog of Life decided that they don't care some name anymore, this information disappears completely. And the reason I did not figure out yet how to keep in perpetuity all the variations from 100 sources as it happens in time. So I try to change sources every half a year. I try to update most important ones. And what I did not figure out how with the budget that we have to keep all this information for every layer in a time layer in perpetuity. So unfortunately, for decision how something is verified, I don't have a solution.
There is a related question. Does the output from GN verifier, for instance, include unique identifier from the checklist sources like IPNI ID?
Yes, it does. And also it creates a link that you can click on and go to mentioned name in that particular source. And if you look at BHL data, they also provide these links for names that are in the particular pages in BHL.
Okay, one more question. Dima, great work, great team. Do you have an estimate of how much of the taxon name strings to taxon concept matching work has been completed for the entire biodiversity in English?
Well, I would say it is completed by definition in the sense that all this data is somewhere, right? If you know about something, it is published, and that's what makes it known. Probably you mean like online, right? So, or like how much of this we are aware of. I would say that like with the catalog of life, for example, gap analysis shows about 500,000 short from what is in all the aggregators, and like 300, 500 300, 500,000 names missing.
Okay. I think that is all for the time being.
Excellent. Thank you, Dima. Okay, our next speaker is Vijay Barve from the Florida Museum of Natural History. His talk is Taxonomy, Compilation, and Curation within R.
Okay, you can see my screen.
Yes.
Okay, so I'm basically going to share some of my recent work in some sense. As we have been discussing, everyone is talking about need for a good taxonomic list to start with. Most of the projects, may it be digitization of specimen, may it be macroecology, or trying to compile trade database or biotic interaction. We need a solid backbone list, and everyone is looking for that. So what has happened is over last 3-4 years, I've been involved with different projects like Butterfly Net, Map of Life, Terrestrial Parasite Tracker, and so on. And I've been developing some scripts, some kind of tools to sort these, clean these, match this list. And then I thought, because there were a few people who were really keen to use them, or basically making it possible to share, I thought I will put this into a R package. I call it Taxo Tools. So it's basically a toolbox for small or medium projects. I've tested it with the World Flora Online. So up to 1 million or a little more than that names, it works quite all right on a simple laptop. So what it has basically is set of functions to do different tasks that we routinely need to do. So unfortunately, I did not really start with a data standard format. Because my lists were in a particular format, I started using that. My pet name for that is just a taxo list format. It basically has a name, some additional fields like higher taxonomy and sources and things like that. And these names are managed using the IDs and the accepted IDs. So basically, every name has a unique ID. And if it is not an accepted name, if it is a synonym of some type, then it has the ID of the accepted name in the accepted ID column. That's how it's a simple list. That is how I maintain. The advantage of this is all the names are in the same bin, the same canonical name column, and then it's very easy to basically run queries on it or match it with different things. So I have different functions. I have some name harvesting functions to get names from ITIS or Wikipedia. Wikipedia has this nice taxo box, and that has many times synonyms listed. So I'm basically trying to just query all the Wikipedia pages of your taxonomic list and pull all those synonyms to build a table. So these are the name harvesting tools, so to say. And then I have some name manipulation tables. It's not as powerful as what Dima was just explaining, but just some rudimentary things like breaking canonical names into genus, species, and subspecies, and vice versa. Or sometimes you have like accepted name and several synonyms separated by either a comma or a semicolon or something, and you want them into separate lines. So some really basic simple functions to manipulate that and make your list cleaner. And then I have wrapped DMA's GNA tools and a GBIF name parser into this just to quickly check your name list, whether they are making sense or not. And then I also have a pretty powerful function to match the list of names with your own master list, where I have fuzzy matching. I also try to look at the combinations of at the genus level synonyms. So sometimes there is a whole genus described, and then the genus name keeps changing, and the names have different combinations. So I make that combinations and check whether if it is missing. Of course, all these, if the names are not directly matching, then sometimes just elevating subspecies to species level, if that data is not available in the master list. Checking if by mistake someone has written just a genus or family rather than a complete scientific name. And then I also have a facility to plug in a user-defined lookup table. Basically, if you know that there are some problems and you can fix them, and then repeatedly running, you can just plug that in saying, okay, if this name comes, this is what I mean, that kind of thing. This is a pretty powerful name matching function. Then there are some list processing functions, basically to tidy up your list. So the building the genus level synonym table so that we can plug in it into the earlier searches, getting higher taxonomic rank data. Sometimes people don't want to deal with subspecies. They just want to treat them as synonyms at the species level. So generating that list, the synonymizing all the subspecies, or trying to guess the taxonomic rank or manipulate these ID numbers. If you have like subsetting them, then kind of compressing all these ID numbers. List matching functions are basically to match to list. You have a master list, you have a checklist. You want to see whether all the entities in checklist are there in the master list or not. Also find some basic statistics like Jaccard index on these lists. Merging list again is a powerful function. If you have 2 or 3 major sources of master list and you want to merge them, you can use this function and basically generate these this merge list together. I have an example here. So you have two lists on the left. So you have some names in the first list, you have a few other names in the other list, and you do have the linkages as specified. So when you merge them, it basically puts all together. It gives you which names were the original names, which names were missing and you need to add. And it also gives possible new names that they did not match with anything that you probably need to add to your master list. So this is a function to basically handle that kind of merging to generate bigger master list from all the list of names that you have. And then also have a few functions to help convert between the formats. Now GBIF and ITs give you a kind of Darwin core format data download of certain checklists. So you can use that to quickly get to taxo list to do other operations, or convert your taxo list into Darwin core format. Also generate some kind of synonym list where you have like a accepted name and synonyms separated by commas or things like that. Handling Wikipedia harvested list, converting them into Darwin core and things like that. Export your data for collection management softwares like Symbiota or Emu, and also generate a document-like format. So this is actually format, a kind of document generated through the function, basically listing all the names, what are the synonyms, color coding it. It's quite interesting function. I'm still working on the functionality, but it basically, if you generate this output, it is really, really simple for a taxonomist to look at this list rather than trying to look at a CSV or a Excel table. So they can basically quickly mark what names they need to be added. So in this particular example, all the names in blue were not in the master list, and they tell from which source they have come from, and the researcher can quickly mark saying, yes, I want this, or no, I don't want this, and then that can be implemented. So these are basically some very basic functions that have been kind of wrapping into this package. It's still under development. So the package is available on CRAN as well as on GitHub. If you want to try it out, if you have any suggestions, you are most welcome to reach me through the issues pages. I think that's what I wanted to share with you all, a very simple kind of workbench for our users to manage these lists. And I would like to thank all the people who have been giving me a lot of suggestions, at times code snippets, helping me improve the package, and basically who are the users of these, and also the organizations for whom while working, I learned and put all these functions together. Thank you.
Thank you, Vijay. We have any questions for Vijay?
Not for Vijay, not yet.
Vijay, are you working on a next version?
Yes, I still have a few bug fixes that I need to attend to, and I'm also working basically on making the list to document function more powerful, because several taxonomists have shown interest, saying that format really gives them a simple format to work on, with like paper and pen, and start marking things, because otherwise it's difficult to look at a table and saying, okay, this is linking to that kind of thing. So that is the, yeah, that's what basically I'm working on right now.
Sweet.
We did get a question, if you want to handle that. What kind of Wikipedia editor, what can Wikipedia editors do to help you do your work? I don't think editors realize the synonym section of the taxo box can be used this way.
Yes, basically, like all the Wikipedia editors do, if you see any problem, fix it. That's it. I mean, add any prominent synonyms that you are aware of into the taxo box, and if there are any wrong names by chance, fix it. Try to put a reference to that, because many names I see in those boxes, they don't really have a reference, but you can definitely put a reference saying, okay, I referred this source, and according to that, these two are synonyms. That makes it easier for anyone who is looking at those names to kind of quickly verify.
Excellent. Okay, well, we are past our time a little here, so we'll move on. Our next speaker is Nicolas Dowdy, postdoctoral fellow at Milwaukee Public Museum, and his talk is Taxo Tracker, a collaborative platform for taxonomic resource maintenance.
Okay, thanks. Can you hear me okay?
Yes.
Okay, great. Yeah, so today I'm going to talk about this project that I've been working on for just a short amount of time. This is really just a proof of concept sort of project, and it's called Taxo Tracker. And the inspiration, or sort of the inciting incident that caused me to think about this was, well, in part, this paper here and many others like it, where we're getting a lot of new species described. In this case, it's by DNA barcoding. And in this case, it was like 400 new species of parasitic wasp. And this is a really contentious paper in the entomological world. And so it brought up this idea that even if you have a validly published name, it may not necessarily be accepted or be used by the community. And so I think we have a need to be able to accommodate alternative taxonomic viewpoints. And to address that, I think a good way of getting to that would be to gather metadata around new names. And that would be things like what types of evidence were used to mint those names, in this case, DNA barcode data. And how well supported are those taxa within the community of experts? So ultimately, can we provide information around how taxon concepts are minted and how a huge number of taxonomic rearrangements, new species, new genera, etc. And here are some examples of some papers where just a huge number of taxonomic changes are happening in just a single publication today. And so we really need a paper trail around this process. So generally we're pretty good, I think, at capturing who, what, and where changes, data about changes. So that'd be authors, what change is made, and maybe what publication it happened in. But the how and why, in other words, by what evidence those changes are being made, I think we're not as good at capturing yet. And I think too, we should be okay with embracing that in some cases, we might be uncertain about contentious species or contentious generic assignments. And in my opinion, I think the future of taxonomy really has to be agile to deal with these quick changes, and it needs to be transparent about the process by which these changes are made. So I started working on Taxa Tracker that attempts to at least partially address this problem. And on the one hand, and Teresa and I definitely have had this conversation a lot, but sometimes, part of my mind is we have too many tools, but at the same time, we need more tools. So this is yet another tool. And I just want to touch on 5 main functions of the tool. So the first is to produce centralized, searchable, and easily obtained resources that come in useful formats, which we've heard about a lot already today, which is great. Second, community-driven and critically citation-based suggestions or edits. Third, consensus-driven curatorial decisions. Fourth, expert-reviewed curatorial recommendations to help people in the museum, for example. And finally, effort tracking and getting credit for both suggesters and the reviewers of those suggestions. So the first point, centralized, searchable, and easily obtained resources. This one is pretty straightforward. So we already know about flat CSV files in Darwin Core and other formats. We already know about APIs. But I think one of the unique ones is formatted taxonomic lists of names. And so that's what Vijay was just talking about. So it's great to have that introduction. Here's a small example of such a list. And these really can be helpful for curators and collections who may just want to print a useful list and go walk into the collection and do some curation, move things around, or give it to a student, perhaps. But the second thing, community-driven citation-based curatorial suggestions. So what do I mean by that? So here's like an example taxon page. And for different fields, there can be an edit button. And any member of the community can click that button and be taken to a unique submission form for the type of field that they're trying to suggest an edit or an addition or whatever it may be to that field. And in that process of making the submission, we can capture who's making that submission, when did they make the submission, what information are they submitting? Is it adding a new synonym, adding a new subspecies, editing the description, changing the type locality, etc. And then importantly, the addition of evidence that supports that submitted claim. So adding some kind of citation like, I read it in this paper, and you can add a link in a field. And then the next step to that process is to create a consensus-driven curatorial decision. So once that submission has been made, it gets passed to a different type of user in the system, someone who has some curatorial expertise. And that's kind of a vague definition. In my mind, it doesn't necessarily mean somebody who is a curator at a museum or something like that, but rather just anyone with some domain expertise on that subject. And then the curators take the submissions and they can review those submissions. They can check the evidence that was provided by the submitter for that claim. And then they can either accept it, reject it, or they can remain undecided about it. And the final part of making or completing their review would be to add just a short description of how and why that decision was made. And really important, I think, is that we can also have one or more curators weigh in on a single suggestion. And so we can get disagreement between data curators, or we can remain uncertain about certain types of submissions. And I think through that process, we can really improve our understanding about the strength of consensus about a particular piece of information, whether that be a name, whether a name is a synonym or not, or even down to particular pieces of data like the type locality or whatever. And we can also start to gather together what the supporting evidence is for these various claims, whether that be a reference or something like that. And this is just an example of how we might display these curatorial recommendations that have gone through a review process. So we can have verified information, which denotes strong aligned consensus among curators. We can have unsupported information, which would be strong counter consensus to a claim. We can have contested information, which would demonstrate weak consensus one way or the other. And then unverified information, which would be information that hasn't been assessed yet, or where curators just remain uncertain about that. And I think one great thing about this is that we can then take that information and push it to curators in a museum, for example, to say, okay, based on the information that we have, we believe you can confidently move your specimens that are currently curated as, in this case, Helicodota trigona into Bertholdia trigona. There's strong consensus for that synonym. Alternatively, we have this other case where we're uncertain or there's contested information. In that case, we might recommend that you conservatively leave this Automolus trigona as it is until we have better consensus around that piece of data. And the last aim is this effort tracking and credit for suggestions and reviews. So I think that credit for the efforts behind building all of these linkages is really, really, really important. So I think submitters, they need credit for making submissions, and curators need credit for their curation. And really, I think David Shorehouse's Bionomia is a huge inspiration for me on that. I don't think I probably would have thought of that without that project. So please go check it out. It seeks to do something similar, but around sort of connecting people to collecting events and specimens that they've collected and identified. So as an overview, there's kind of these three pieces, and I think you could sort of think of them similar to how we think of preprints, peer review, and final publication. So on the preprint side, the generation of new information, we have community provided suggestions or even curator initiated updates to the resource. And we can also add to this imported data from external sources, maybe Plazi or GBIF or something like that. And then there's this kind of big filtration point at the curator driven verification. And through that process, we can create our knowledge outputs, which generates verified information. It can generate the community credit for suggestors and for curators. The overall vetting process for information, I think, becomes at least a little bit more clear. And we can start to assess the community's confidence in names and data associated with taxa, and maybe even start to begin explicitly defining what taxon or taxonomic concepts are, which I think often are not really well defined in the literature. Of course, there's still challenges behind a system like this. Probably the number one challenge is you need curators to do this. So curators are certainly required. But as part of this process, I think we also get to learn where expertise is missing. So what taxonomic groups do we not have people with domain expertise? Which groups do we have a lot of expertise in? And the other challenges are, I think, without that credit, especially for the users who are making the suggestions, they need to understand the value of those credits. And also curators, and more so maybe the administration that pays those curators, needs to understand the value added by doing this type of work. So without community and curator buy-in, I think this really can't happen in a meaningful way. I think there's some other fun features. So you could think about, creating watched lists at arbitrary taxonomic levels. So if my collection has a lot of bombus and I want to get like push notifications to my email when changes to any bombus species are either suggested or the review process has completed, you could do that. Or if you have a list of what taxa are in your collection, you could create a watched list for those taxa and sort of get updates as they come through. You could have suggestion and curation leaderboards. And I think another cool thing would be to have a process by which you could suggest suggesters. So if you know that users X, Y, and Z have suggested data for various species of Cicindela, maybe you could ping those people and say, hey, we've got some unverified information for this species in this genus that you've worked on. Maybe you could help us fill in some information here. And I'd love to hear some more ideas on other things we could do. With that, I'd like to thank you all for listening. I really want to thank the Terrestrial Parasite Tracker project because that really funded. I spent about a month or two on this project while I was on the TPT project. So this would not have happened without TPT. I also want to thank my co-organizers, Vijay and Teresa, who really gave me a lot of feedback and input on this project. And there's way too many projects out there and tools and resources that I took inspiration from. So I thank all of those developers. And of course, I thank Tadwig for putting this conference together.
Thanks, Nick. Do we have questions for Nick?
Yes, we do.
Imagine that.
So, yeah. First question is the reasoning that expert curators use to react to consensus decisions recorded in the system? Is the final evidence for a claim is recorded?
I think I didn't catch that whole question.
Okay, you want me to
Oh, I
read
see
again?
it here. I can see it here. Yes, so the answer is yes. So we record all the pieces of information associated with decisions that are made about the verification of any claim. So what curators, made a decision, how they came about, how they came to that decision, but then also what another curator perhaps said about that same piece of information. And those things could conflict, for example, or they could both agree or whatever.
Okay, one more question. Any idea how to share and propagate curatorial recommendations, W3C web annotations maybe? This from David Charters.
Yeah, I think that is definitely something that I want to look into, how we would have this system where we're building consensus around things, but we need to be able to put that back out to other resources, especially, we might be getting new names coming into the system that may not be in other places yet. And so, yeah, I don't know what that process is right now because I haven't built that part yet, but I definitely want to do that.
That's all I think. We have a couple of questions for earlier presenters, but that will take after the final presentation.
All right, thanks.
Great. Okay. So I'm the next speaker. My name is Teresa Mayfield-Meyer, and I am the community coordinator for Arctos. My talk is using the taxonomic backbones, the challenge of selecting a taxonomic resource and integrating it with a collection management solution. So I am coming at this a bit, get my screen shared here, from the consumer or user perspective. So maybe a little bit different than all of the other presentations. And I'm going to be talking about taxonomy from an almost non-biologist point of view. So hang on with me. The reality is there is not a single taxonomic backbone. There are a bunch of somewhat interconnected taxonomic nodes. The Global Biodiversity Information Facility, or GBIF backbone taxonomy, the World Register of Marine Species, or WORMS, and Mollusca base, to name a few. While a unified classification backbone is what's needed, what we have is more like a tangled web of taxonomic information, often with some authorities nested within others. Mollusca base contributes to WORMS, which contributes to Catalog of Life, which contributes to the GBIF backbone taxonomy. So, how is a collection manager without expertise in a given set of taxa and a limited amount of time devoted to finding the most current taxonomy supposed to maintain research grade identifications when there are so many seemingly authoritative taxonomic resources? And once a resource is chosen, how can they seamlessly use the information in that resource? The Arctos community has attempted to make managing taxonomy easier by sharing taxonomic names and their associated classifications across 44 institutions holding over 230 collections. This helps spread out the work of maintaining current taxonomic information, but it can only work as well as the expertise and time available. Arctos also includes taxonomic information from a variety of sources accessed through global names. The global names architecture is a system of web services which helps people to register, find, index, check, and organize scientific names and interconnect online information about species. This we saw in Dima's presentation. Classifications found in the Arctos external resources are primarily created from information ingested from global names and can be cloned into Arctos, providing easy access to externally managed taxonomic resources on a name-by-name basis. Here's what the Arctos programmer, Dusty McDonald, says about global names. Quote, use it. It's why they exist and they're freaking awesome. We're already talking to them, so the next new source that shows up there will just magically appear in Arctos. Anything we do to improve some existing source accidentally improves everything else as well, and their API is very simple to talk to, so making those improvements is generally trivial. Calling them the biggest thing to ever happen in the taxonomy informatics community doesn't at all feel like a stretch to me, end quote. While global names has proved to be an excellent source of taxonomic information that is easy to use, there are a few drawbacks. The first is that updates from primary sources can be sporadic for many reasons, including that they require manipulation before ingestion into global names. Here we see an example of this in a taxon that's been added to WORMS, but has not yet made it to global names. We heard about global names ingestion of resources in Dima's talk. Another drawback is that not all of the information from primary resources is ingested in global names. We get much richer data directly from WORMS, but it comes at a cost. Another quote from Dusty about WORMS. WORMS's API is pretty nice, but it's also fairly complicated. Even if all of global names sources had an API, they don't. And even if all of those APIs were about as nice as WORMS, they're not. It would be something over 194 times the work to develop direct connections, end quote. Have I mentioned that our programmer really likes global names? This points to a need for community-wide support of such resources as global names. What would it take to help global names create direct connections with primary sources or somehow stay more current and add needed functionality? Who else is making use of this resource? And can we get a community behind global names to make them even more awesome? And after seeing Nick's talk, I feel like there's some synergies between those two efforts that would be even better. Now, back to using the backbone. Arctos has developed a hybrid model for using worms taxonomy. We still access worms data via global names, but for a certain subset of names, we directly access the worms API and ingest the taxon metadata, relationships, and classification. This process is resource intensive, and we are constantly looking at ways to minimize the need for processing. But it still often ends up in requests for manual refreshes of segments of the worms via Arctos data. This is not ideal, but it's been a big step forward in taxonomy maintenance for some Arctos collections. This is Phyllis Sharp, the shepherd of worms via Arctos. She's going to describe what access to this taxonomic resource has done for her and her team of digitization volunteers in the Denver Museum of Nature and Science Marine Invertebrates collection.
I'm Phyllis Sharp, and a volunteer departmental associate working with the Marine Invertebrate Collection at the Denver Museum of Nature and Science. None of the volunteers who do the day-to-day work in this collection are database or taxonomic professionals, so we have always relied on external sources that tap into global experts. For years, finding, verifying, and updating taxon names and classifications was time-consuming and prone to errors. All that changed for us with the World Register of Marine Species, or WORMS. The taxonomic editors at WORMS represent the best of the subject experts on all marine phyla. Additionally, they are adding freshwater, terrestrial, and fossil mollusks that are curated by Molluscabase. It's like having an expert on staff and available 24/7. WORMS assigns each taxon name a unique APIA ID. Arctos added the WORMS database as a built-in taxonomic source, using this APIA ID to automatically populate and refresh the taxon names, classification, and metadata. This has reduced the time spent managing taxonomy by at least 90%. Using the names built into WORMS via Arctos almost eliminates typographical errors during data entry and helps us avoid spreading nonsensical combinations in our historic records. Using an externally managed taxonomic source does require some adjustments. Changes in the acceptability of a taxon name occur frequently, and our goal is to update to the currently accepted taxon names. Arctos has a report that lists the taxa we are using and current identifications that are unaccepted by WORMS. Using the WORMS match tool, we can easily find the accepted taxon name and add it as the current identification. If our collection had location identifiers, that would be the end of the effort, but we are organized by family and species. So we do relabel these lots with the new taxon names so they match the current ID in Arctos. This also assures that lots recently databased with the current classification are stored with those databased prior to the change. Dealing with changes in higher classification requires another adjustment. We may not be aware of changes in the family that a species is assigned to. For example, when the subfamily Sematinae in the family Rhinellidae was elevated to family status, it automatically changed the family in the WORMS via Arctos classification. So we did relabel and relocate those lots. We also keep an extensive list of all the families in the WORMS taxon tree and periodically update it to find changes. Not every taxon name is yet in WORMS, necessitating a broader search. Good sources include the taxonomic backbone at GBIF and at iDigBio. With both, we can search for species within a defined geographic area that are in use by other museums. We also use Google Scholar and reputable shale dealer websites to track down synonyms that might lead us to the currently accepted name. If we can't find any reputable source for a taxon name, Arctos enables us to enter a string to describe the specimen without generating an invalid taxon name and classification. When using these other sites, we are aware that we may be the source of the lots that reinforce the validity of the taxon name. We don't want to breathe our own exhaust or spread invalid taxon names into the cloud. Having WORMS as the taxonomic source for the majority of our specimens has greatly increased the quality of the data we provide. I'll be here after the presentation to answer any questions.
So Phyllis can't be here, but if you have questions for her, I can pass them on.
I'm Phyllis Sharp, and
Oh, sorry. So now to wrap up, this quote from Stephen Garnett really hit home for me. Currently, no single agreed list of species is available. Instead, some iconic groups of organisms, such as mammals and birds, have several competing lists, while other, less well-known groups have none. The none is a real problem, but do we need an agreed list? I'm not sure we do, and I think Nick may have proved that. What we need is a comprehensive list so that we can select names based upon our needs, whether keeping current with changing nomenclature or a specific authority, or maintaining names that reflect the physical organization of our collections. This may be nuts, and it's the opinion of someone without a degree in biology, but until we build a resource for biological taxon names that includes metadata about them, such as who published them and when, where the type can be located, and all the relationships they have to other names, we will continue to have a tangled web of taxonomy that stymies research and creates public distrust in science. In the meantime, we do have global names to help find resources and resolve names, and for that I am grateful. Just look out for your own exhaust. Thanks to everyone who contributed to this effort, and here's to making taxonomy even better going forward.
All right, thanks, Teresa. We do have a question for you. That question is, you mentioned in passing that collection management systems should more cleanly separate taxon names from objects like specimen determinations. Can you elaborate?
Well, so yes, I guess, not being a taxonomist, I can do my best. So it's one thing to have a taxon name. It's another thing to say, I looked at an object and I agree that it belongs as part of this taxon name. So in a CMS, in my opinion, you need to have the information about who assigned a taxon name to the objects that are being described.
We have another question. So it's it's a multi-partner. I'll read the first part. Can you clarify how researchers in Arctos maintain custom name lists? Is there a tool within Arctos?
Yes, so we do maintain taxonomy locally. And so we have a very series of tables that go together for managing taxonomy. A lot of the taxonomy that is currently managed within Arctos was imported, some of it almost 20 years ago, and is being maintained by whomever is using it. So yes, we do maintain that internally.
Okay, second part to that question. Also, what is the pathway suggest to Arctos users right now to get these custom lists into global names?
Oh, to feed them back the other way. So we haven't really talked about that. I'm sure we could probably pretty easily just work with DEMA to figure out the best way to do that. The thing is, for now anyway, I think we maybe only have one custom list that didn't already sort of come to us from global names in the first place. We have several custom lists within Arctos, and new ones get created quite often. But we also have kind of the original master list called the Arctos taxonomy. And a lot of that came from imports of data many years ago. So there have been changes to it, and it could be that, feeding that back into global names would make sense at this point.
I want to add that global names has a in and out connection to Arctos. So every half a year, we update Arctos as well, and have everything that is new in Arctos in global names. So if there is a checklist that is in Arctos, it will be in global names. And thank you so much for all your nice words about Gian.
Well, Dusty just can't say enough about how easy it is to work with you. So,
yay.
We have time for one more. I think we have one more question for you. Okay. Teresa, can you clarify? Oh, I'm sorry. That is not the right question. There it is. Okay. Teresa, what I think they mean, would Arctos be severely broken if taxonomy, i.e. an editing interface, were completely removed and entirely reliant on external services?
No, it would not. There are parts that immediately would not work, but could be easily fixed. So the way those external sources work within Arctos is they facilitate searching. So if I come in and search on a name that's in one of those sources, I will find records in Arctos that are related to that name. So if all of our internal taxonomy went away, you could still search in Arctos based on taxonomy using those external sources. The only thing it would do is, so in a given catalog record, when you look at an identification, you see a taxon name with a classification under it. And that classification comes from the collection's preferred resource for taxonomy, which is one of the internal local sources. So that, you know, what you would see on a catalog record page would be gone for until we figured out how to just use the external sources, which is something we've been talking about anyway, potentially just letting people use those external sources as a preferred taxonomy.
All right, I think that's it. Thank you.
Great. So do we have general questions now that?
I think we have a few questions lingering for Dima, actually. Maybe we can handle those now.
Sure.
So Dima, the question is, what would it take to make GN verifier near real time with, for example, catalog of life checklist bank?
This is a goal, definitely. I would say it depends on availability of the source. For example, for some sources, I have to write an email. Then the person sends me a file. Usually this file is not consistent with how it was before. And I have to redo the things, like to make it work again. But for many sources, like for example, for Arctos now, we do have a much easier system. So there is a Arctos dump that I can access anytime I want and get data from it. So I would say it is for sources that are like Arctos, I think it is possible to make changes, maybe not real time, but like every day, probably. I would say for that, what I need is a new harvester. I still use harvester I wrote in Ruby, and it is way slower than like, you know, what it can be. So this is pretty much a question of a new harvester. And if when it exists, I think we can get to maybe every day for some sources.
We have one more question. This is for Dima or anyone, so maybe something to think about generally. This is from Katja Seltzman. I appreciate all of the work managing taxonomic names, and I see the tools being developed as very useful. I recently used the tax size package, and it sounds like it uses global names as a backbone. So my question is, if I have my own taxon names list, how best or easiest is it for me to include my name list into global names services?
Write an email to me. I will include it, and it will be there. There are some sources that are included this way recently, like Miriatrix, for example, from Carlos, and yeah, that's definitely possible. and format can be simple. It's the simplest possible is UID and a name. Like if there is a classification, it's better. If there are accepted names, it's even better. And I will, the result will be going back to your data if it is online.
Carlos had a comment to make, right?
Yeah, but so maybe Dima, you remember that a bit more than 1 year ago, we were working on a missing name that was in the MCC base. So from Harvard, and MCC base uses Arctos. So what I'm wondering is if there are custom lists being made at the institutional level in local Arctos databases that are still not flowing to you, and if it will help to have a general recommendation within the Arctos network that if there are local custom databases at the institutional level, that the curators could make those lists flow to you as well. Because we found at least one name last year that was not in any other database. So maybe those local databases could help you.
So we find that too. So right now in the world, there are 2 instances of Arctos. So one is the big community that I spoke of, and the other is Harvard. They run their own instance. So probably, I think it's just maybe us connecting Dima with them so that he can do the same thing with them that he does with us.
Harvard is in already.
Oh, okay. So
Exactly because our conversations.
Okay.
yeah.
Okay, that's done then from May last year. Okay, that's good. And then the other thing was that so I posted the link in this chat. Maybe I should also post it in the WOWA chat. But there is a special issue on this sort of towards a global list of accepted species. And I have the same question that Teresa, that if we want that, but I will say that people should go read those 5 papers. There is one more that's still not being published, but I posted the link to the special volume. So when the 6th paper is out, you will find it with the same link, and I will post the same link in the WOWA chat. But the thing is that that's not how taxonomy works, having one global list of accepted names. And we should, I'm telling this to myself because now I'm also managing a list of names attached to a classification. Yeah, and classifications are hypotheses.
Yep.
And the names themselves are hypotheses. So we have an ethical duty also to hear the voices of the minorities. Yeah, because this is not as simple as being right or wrong, and that my classification is right, and that my generic placement for this group of species is right. Yeah, all those things are hypotheses. And of course, users need a yes or no solution, but we should be very clear and very ethical on what we do and what we represent, and also be conscious that when we represent only one classification, because technology doesn't allow us for more at its current state, we also have an ethical duty to inform on potential misuses. Yeah.
And I'd like to add to that the whole idea of I know it's part of what Phyllis said, about finding the accepted or correct. But I think we need, as a community, to help educate the rest of the world that what's accepted today may not be accepted tomorrow, that this is all in flux, and you can't ever expect really to get the right answer because life is evolving and changing, and our knowledge about it is evolving and changing. And so, as you say, it's much better to have all the competing ideas so that you can evaluate them rather than just knowing what's right.
So for example, if to take like catalog of life, right? So I think it is really great that they have only one opinion, because otherwise it would be for like ecology people, for governments, go and see like 50 different opinions about something, and how to make policy from that. So I think it is great that catalog of life has only one opinion, but I think it would be really great also if like there is a clade, and let's say there is a TaxonWorks page that alternative group developed and maintained, and they say, okay, this is a opinion from catalog of life. If this is not what you're looking for, there is this TaxonWorks page, this is that TaxonWorks page, and they can be competing. And this way we have both worlds. We have multiple opinions, and we have like a sort of consensus that is good enough for like European governments, for example.
Right.
I think it would be good if we at some point have software that allow us to display multiple alternative classifications, which, for example, Scratchpads doesn't have. So I can just display one classification. And the other thing is that we also need the same software displaying classifications over time.
Mm-hmm.
Because if, for example, it would be really cool if we could add the time dimension, and then I could go back in a simple step and a simple interface to what the classification was in 1900. Yeah.
So this goes to what Yorit was saying.
Yeah, we're missing the alternative classifications, and we are missing the temporal dimension. So any development towards this will be great, at least from my perspective.
Yeah.
I was totally.
Oh, sorry. I just wanted to jump in to say that that's why I wanted to create the tool for assembling taxonomies, because I want people to be able to push old classifications online so that you can track the history of the classifications for particular taxa that you're interested in, because that's not possibly right now. Platzi does a lot of that, but only for things that are available digitally. And we know that's not the case for a lot of taxa. So yes, +1 for multiple taxonomies available.
So if you look at that big hairball of relationships from that nomenclatural data that I presented, that has all of the chronological data that you need. So someone like Laura, who's got some creative ways of, or Debbie has been proposing this, I want to see the timeline version. In fact, the interfaces that we present in TaxonWorks are actually timeline versions. They're just linearized. But if people want real data to work with in that visualization challenge, then data sets like DEMAs that are super rich like that, they're there waiting for you to visualize with all that time data that you want.
I was really shocked when I added to global names index animalian from Sherborne, and I found that 25% of names that are there, zoological names, they just gone. They are not in any data source. They're only in index animalium from 19th century.
Mm-hmm.
25%.
Yeah. So that's a lot of history lost, right?
Yeah.
All right, well, we've actually gone a little bit over time. I really appreciate everybody being here. All of the presentations were great. I've learned a lot, and I hope we can keep this conversation going and maybe work together more as a community instead of everybody in their little silos doing their thing, because I think that's been taxonomy's biggest problem from the beginning of taxonomy. Let's see if we can keep talking to each other. And thanks everyone for being here. Thanks to Tadwig for hosting this symposium.
Thanks, everybody.
Thanks, organizers, for putting this together and getting everybody together.
Thanks so much, Teresa and Vijay and everybody for moderating. We are taking about a 30-minute break. Up next, we are going to have kind of an informal poster session, lightning talks. It'll be pretty informal, but if you have any ideas or you just want to come to the table with something, feel free to unmute yourselves in about 30 minutes. And if you need any more information, you can reach out to Deb and Gail or some of the other organizers within the Whova messages or in the chat function. So we'll see you then.